You might have read that there are a whole set of different strains of SARS-CoV-2 viruses circling the globe, each with a different associated level of danger. In this episode of Shareable Science Beyond the Blog, we'll look at the science and see if it matches up to that claim. Welcome to Shareable Science. Science you can share. We're going to start this video at the bottom of the whiteboard. This is a great reference if you want to know more about how viruses mutate and the meaning of those mutations and how we analyze those. Much more information than we're going to be able to cover in this video. Okay, back up to the top. The SARS-CoV-2 virus is what's called an RNA genome virus. That means that its genetic information is made of RNA, not DNA like you find inside the genomes of our cells. Specifically, the SARS-CoV-2 virus contains just about 30,000 letters of genetic information. And here's a schematic of what that genome looks like, how those 30,000 letters are broken out. Each one of these boxes represents a gene, or in some cases a set of genes. And remember, genes provide instructions for how the virus, in this case, needs to make the proteins to do its job. Those boxes that are in color represent genes that tell the body, the cell, how to make virus proteins that are important in the structure of the virus. For example, the blue box is the spike gene. It provides instructions for how to make the spike protein that sits on the outside of the SARS-CoV-2 virus. In general, RNA viruses mutate really rapidly. Most often, they don't have any kind of proofreading capability. So every time they make a copy of their genome, and viruses make lots and lots of copies of their genome to make lots and lots of viruses, every time that copying gets made, there's a risk that you're going to introduce an error, usually single letter changes in the recipe. The SARS-CoV-2 virus, however, does have a little bit of proofreading capabilities, so it still introduces errors, but at a generally slower rate. In fact, a recent study looking at virus samples from 90,000 patients and sequencing through that set of instructions found about 12,000 different mutations across that 90,000 sample. So those are mostly single letter changes. And on average, any two SARS-CoV-2 viruses will have about 10 differences between them. The key question is, what's the importance of those differences? Are they even important to begin with? Do they have some sort of clinical relevance? And to dig deeper into that, let's go play with some models. So let's play with Legos for a minute. Now, here's how this model works. There's a set of instructions, usually a pretty complicated set of instructions, that tell you which Lego pieces get built together in order to make your model. This is your genome. This provides the instructions. And this is the finished product. These are your sets of proteins. Now, in reality, this is not what your proteins look like. They actually look more like this, where each one of these is an amino acid, and the instructions tell you how to put the amino acid together in a specific order that gives you structure and meaning. So we're going to set this aside for the moment and work with our cubed blocks. Now I've got two of these. Let's say that this is the set of virus proteins that's built with the normal or the typical set of instructions, the typical genome. And let's say that this is the one where we have mutation introduced. So we can do some comparison between the two. So right off the bat, we've changed our instructions, and this is what we're going to compare against. And in some cases, you might not even be able to tell any difference at all. This looks exactly the same. But the reality is, it might be something as straightforward as I've substituted 2 4s for 1 8. That's a genetic change that has no impact whatsoever. I might have other changes that are a little bit easier to see, and you can see those from the different colors, or maybe even the deletion, the missing piece. Again, this doesn't really change the structure. It does change the way it looks, but it doesn't change the way that this behaves. All right, both of these that look exactly the same and these that look a little bit different but don't change the overall structure, these are variants. These are not new strains of the virus. Let's get that terminology really carefully set at this point. So the vast majority of those 12,000 DNA changes that I told you about don't lead to new strains. They're just variants within one strain because they don't change the way that it functions. Now, what would we need to see in order to think about calling something a completely different strain? Let's look at this. So in this case, there's a pretty big change that creates a completely new function, a completely new physical property or clinical property. Maybe it actually makes the virus more likely to cause serious illness, or maybe it makes the virus more likely to be resistant to a certain medication. If you had a genetic change that led to something like this, this would now become a completely different strain of the virus. A great example, stepping away from coronavirus, is influenza. So mutations happen all the time in influenza viruses, and some of them mean that even if we've had the flu before, the new mutations create changes in the proteins on the outside of the virus so that our immune system doesn't recognize having been infected before. Those are completely new strains because they've added a new biological property. It's one of the reasons why you have to get a new flu shot year after year. That would be a completely different strain. Okay, back to our coronavirus. Sometimes you actually see differences like this, where we don't really know what this means. It looks different, it sticks out from the wall, it has a different structure, but does it actually have a different function? And so how do you tell the difference? How do you determine if this is really more like this or is actually a different strain? So a great example of that is a mutation called D614G. It's one you may have heard about before. It's specifically in the gene that codes for the spike protein, and it changes the 614th amino acid. It was seen early on, maybe in March, in different places around the world, and by June, it had become the dominant form of the virus. So almost every analysis, every sample taken since June contains that specific mutation, that specific change. Additional work in the lab suggests that this change actually leads to a virus that transmits a little bit more effectively. It spreads a little bit faster. It doesn't seem to change the clinical outcome. Patients with the original version and patients with the mutation don't seem to have any difference in how sick they get or their likelihood of recovering. But that change is enough of a physical difference that potentially it really gets classified as a strain, not just as a variant. So we probably have 2 strains of the SARS-CoV-2 virus circulating right now. Not 6, not 8, not a dozen. As we discover more variation, it's likely that we'll find additional changes that have important properties. But again, so far, nothing has been found that has a clinical impact that makes the virus worse or better or less resistant or more sensitive to a vaccine. That's a lot of content and a lot of definitions talking about how changes in instructions actually lead to unimportant or maybe important differences in the virus. I hope you find this useful. If you think other people might benefit from this kind of a conversation about what is a variant versus what is an actual strain, please share this video. Thank you for watching, and I look forward to seeing you next time on Shareable Science Beyond the Blog.